Our brains are incredibly complex organs. They allow us to think, feel, and experience the world around us. The answer lies in tiny cells called neurons. These neurons are the building blocks of the nervous system, transmitting information throughout our bodies. Imagine them as messengers, constantly sending and receiving electrical and chemical signals. These signals dictate everything from our senses to our movements. Get ready to unlock the secrets of neuronal communication and gain a deeper understanding of the amazing capabilities of the human brain. Before we dive into action potentials, let's understand electrical potential. Electrical potential is the difference in electrical charge between 2 points. Neurons maintain an electrical potential across their cell membranes. This potential, known as the resting membrane potential, is crucial for neuronal communication. The resting membrane potential is created by an uneven distribution of ions inside and outside the neuron. Think of the neuron as a tiny battery, storing electrical energy that can be released to generate an action potential. This careful regulation of ion movement is essential for maintaining the resting membrane potential and preparing the neuron to fire an action potential. Imagine a neuron as a tiny, bustling city. Inside the city walls, there's a constant flow of ions, crucial for the city's function. This balance in the neuronal world is called the resting membrane potential. It's the electrical difference between the inside and outside of a neuron when it's not sending signals. This potential, typically around -70 millivolts, is a dynamic equilibrium maintained by the movement of ions across the cell membrane. This negative charge acts like a coiled spring, storing potential energy that can be unleashed to generate an electrical signal when the neuron receives a strong enough stimulus. In our cellular city, potassium ions, or K+, are particularly important residents. They prefer to be inside the neuron, contributing to the negative internal environment. These potassium ions move relatively freely across the cell membrane through specialized channels. However, positively charged sodium ions, or Na+, are mostly kept out, creating a concentration gradient. The cell membrane, with its selective permeability, plays the role of a vigilant gatekeeper. It ensures that the neuron is always prepared to fire an action potential when needed. Maintaining this delicate balance of ions requires the sodium-potassium pump. This pump, embedded within the cell membrane, works tirelessly to maintain the resting membrane potential. The sodium-potassium pump uses energy to transport 3 sodium ions out of the neuron for every 2 potassium ions it brings in. This constant exchange of ions ensures that the inside of the neuron remains more negative than the outside. Without this constant maintenance, the resting membrane potential would dissipate, and neurons would be unable to communicate. This would be like our city losing its energy source, bringing all activity to a standstill. Now, let's delve into the electrifying world of the action potential, the neuron's way of transmitting information. Imagine a sudden burst of activity in our cellular city, a wave of excitement sweeping through the streets. This is the action potential, a rapid and transient change in the neuron's membrane potential. When a neuron receives a sufficiently strong signal, it triggers this remarkable event. This threshold, known as the threshold potential, is like a trigger point. Once crossed, there's no turning back. The action potential is initiated. A cascade of events unfolds. Imagine the city gates flung open, a rush of citizens pushing their way into the city center. This is depolarization, the first key stage of the action potential. Voltage-gated sodium channels open up, allowing sodium ions to surge into the neuron. This influx of positive charge causes the membrane potential to become rapidly more positive. The inside of the neuron becomes positively charged compared to the outside, a dramatic shift from the resting state. The action potential doesn't linger. It's a fleeting change, a spike in electrical activity. The influx of sodium ions is short-lived. As the membrane potential reaches its peak positive value, the sodium channels begin to close. This marks the beginning of repolarization, the return to a state of negativity inside the neuron. Potassium channels, which opened more slowly in response to the depolarization, are now fully open. Potassium ions rush out of the neuron, helping to restore the negative membrane potential. The neuron briefly dips below its resting value, a state called hyperpolarization, before returning to its normal rhythm. Now, let's turn our attention to the unsung heroes of this intricate process, the ion channels. These tiny protein pores embedded within the neuron's cell membrane act as gatekeepers, controlling the flow of ions in and out of the cell. These channels are incredibly selective, allowing only certain types of ions to pass through while barring others. This selectivity is crucial for maintaining the resting membrane potential and orchestrating the action potential. Some channels are always open, allowing a constant passive flow of ions. Others, like the voltage-gated channels, are more dynamic, opening and closing in response to specific triggers. Voltage-gated channels are particularly sensitive to changes in the membrane potential. These channels are like highly alert guards, constantly monitoring the city's energy levels and responding accordingly. When the membrane potential reaches a certain threshold, it's as if an alarm bell rings, prompting these channels to spring into action. The opening of voltage-gated sodium channels triggers the depolarization phase of the action potential. These channels are designed to inactivate quickly after opening, preventing a continuous influx of sodium ions. This inactivation is crucial for allowing the neuron to repolarize, to reset itself for the next action potential. While voltage-gated sodium channels are essential for depolarization, voltage-gated potassium channels play a vital role in repolarization. These channels open more slowly than sodium channels in response to depolarization. As the membrane potential reaches its peak positive value, these potassium channels are fully open, allowing potassium ions to flow out of the neuron. This outward flow of positive charge helps to restore the negative internal environment, bringing the city back to its resting state. The precise timing of the opening and closing of these channels is critical for the proper functioning of the neuron. This exquisite control over ion movement allows neurons to generate and propagate electrical signals with remarkable precision. Now let's zoom out and witness how this electrical message travels along the neuron's long, slender axon. Imagine a line of dominoes perfectly spaced, poised to topple with the slightest touch. The action potential, once initiated at the axon hillock, sets off a chain reaction, a wave of depolarization that spreads down the axon. This propagation is not a passive flow of electricity like that through a wire. Instead, it's an active process, with each segment of the axon regenerating the action potential anew. This ensures that the action potential travels in one direction, from the axon hillock toward the axon terminal. Nature has devised an ingenious way to enhance the speed of neuronal communication, the myelin sheath. The myelin sheath is a fatty substance that wraps around the axon, forming an insulating layer. This insulation is punctuated by gaps called nodes of Ranvier, where the axon membrane is exposed. These nodes are like booster stations along our cellular highway, maintaining the strength and speed of the signal. The myelin sheath acts as an electrical insulator, preventing leakage of ions across the axon membrane. This insulation allows the depolarization wave to spread rapidly beneath the myelin sheath, jumping from one node of Ranvier to the next, a process known as saltatory conduction. In saltatory conduction, the action potential is regenerated only at the nodes of Ranvier. This leaping of the action potential from node to node significantly increases the speed of conduction. This increased speed is crucial for efficient neuronal communication, especially for signals that need to travel long distances in the nervous system. Myelination, the process of forming the myelin sheath, is essential for the proper development and function of the nervous system. Disruptions in myelination, as seen in diseases like multiple sclerosis, can have severe consequences. The precise and rapid transmission of nerve impulses, facilitated by the myelin sheath, underscores its critical role in our ability to perceive, think, and interact with the world around us. We've delved into the intricate molecular mechanisms of action potentials, the carefully orchestrated movements of ions across cell membranes. But to truly grasp the significance of these electrical impulses, let's step back and explore how they translate into everyday experiences. Imagine a line of dominoes, perfectly arranged, each poised to topple and set off a chain reaction. This simple analogy captures the essence of action potential propagation in neuronal communication. A single neuron receiving a stimulus fires an action potential that travels down its axon, like the first domino falling. This electrical impulse, however, doesn't simply fade away. Upon reaching the axon terminal, it triggers the release of chemical messengers, neurotransmitters, into the synapse, the tiny gap between neurons. These neurotransmitters then bind to receptors on the neighboring neuron, like a signal being passed from one runner to the next in a relay race. This binding can trigger a new action potential in the receiving neuron, continuing the cascade of communication. This process, repeated across billions of neurons in our nervous system, forms the basis of our thoughts, sensations, and actions. Vision is the remarkable ability to perceive the world through light. A single photon strikes a photoreceptor cell in the retina. This triggers a cascade of events leading to our perception of sight. The photoreceptor generates an electrical signal passed to other retinal neurons. These signals are relayed to the optic nerve, carrying visual information to the brain. The optic nerve transmits impulses to the visual cortex. Here, signals are decoded, allowing us to perceive objects. The speed and precision of this process highlight the efficiency of our neuronal communication. We've journeyed deep into the intricate world of neurons, the essence of our thoughts, emotions, and actions. We explored the neuron at rest, its internal environment, and the resting membrane potential. Then, we witnessed the action potential, a rapid shift in electrical charge down the axon. We delved into voltage-gated ion channels and the movement of sodium and potassium ions. The myelin sheath accelerates action potential propagation by enabling it to leap between nodes. Finally, neurotransmitters carry the signal across the synapse, perpetuating neural communication. Understanding the language of neurons, these electrical and chemical signals, is fundamental to unraveling the mysteries of the brain, the most complex organ in the human body. The journey we've embarked upon, while providing a glimpse into the intricacies of neuronal communication, merely scratches the surface of this vast and enigmatic field. As we delve deeper into the workings of the brain, we are constantly amazed by its complexity, its ability to process information, form memories, and generate consciousness. The study of neurons and their communication is at the forefront of neuroscience research, with implications for understanding and treating a wide range of neurological and psychiatric disorders. From developing new therapies for neurodegenerative diseases like Alzheimer's and Parkinson's, to unraveling the neural basis of consciousness, the possibilities are endless. As we continue to explore the intricate workings of the brain, we gain a deeper appreciation for the remarkable capabilities of these tiny cells, the neurons, that orchestrate the symphony of thought, emotion, and experience that defines what it means to be human.